And now we come to that pivotal team of James Watson and Francis Crick. In 1953, they make the monumental discovery of the actual structure of DNA. They've been working on this, racing against an individual called Linus Pauling and other scientists that are trying to figure it out. One of the key sort of things that came out back in 1991 in James Watson's book is he says that he saw Franklin's photo. So Rosalind Franklin's photo of DNA showed that very clearly it was a double helix structure. That idea, so you see the winding DNA structure that we get to see in pictures, that really is the structure of DNA was shown clearly by this picture of this X-ray diffraction picture. James Watson was able to see it, not because Rosalind Franklin showed it to him, but because her supposed boss, who was kind of her coworker, but she was never really willing to be bossed by him. It gets weird. But anyway, at the end of the day, he saw a picture he shouldn't have seen, and it let him know clearly that this was a double helix. That gave him and Francis Crick part of the key information that they needed to figure out the actual structure of DNA. One of the things that was important about figuring out that structure is the shape of a double helix means that the double helix has two strands, right? And that you read them, for it to be a double helix, you have to read them this way. So you read one strand going one direction, the other strand going the other direction, so that it's like one long message that doubles over onto itself the other way. The way it's talked about specifically, the terminology it's used is a 3 to 5 strand and a 5 to 3 strand. That has to do with which sugar, with which of the carbons in the carbon ring is involved in the bonding for the sugar. Specifically, the structure that they found was this structure. This is what they came up with. They understood that they came to understand that the bases had to be on the inside. The adenine, the thymine, the guanine, and the cytosine had to be on the inside. And that meant that the sugar, the deoxyribose, and the phosphate group that it was attached to had to be on the outside. And so they came up with this model in which there was this sugar phosphate backbone. Phosphate, sugar, phosphate, sugar, phosphate, running on the outside. And then on the inside were the actual bases, the T and the A and the G and the C. On the opposite side, running the other direction, notice how this sugar molecule is upside down compared to this sugar molecule, that these two deoxyribose are headed different directions. Over on this side, you had another sugar phosphate backbone with on the inside the actual bases, the T or the C or the G or the A. Understanding this structure also was contributed to by Chargaff's work. Remember, Chargaff was the guy that figured out that in all living things, the amount of adenine in the DNA is equal to the amount of thymine in the DNA. The amount of guanine in the DNA is equal to the amount of cytosine in the DNA. And basically what he was discovering is that A always pairs with T and G always pairs with C. By understanding that, Watson and Crick were able to construct a model that not only had these bases on the inside, but showed the way that these hydrogen bonds between the two sides lined up so that the two sides could stay bonded together, but could split apart for the purpose of communicating this genetic information, this genetic code, and then easily be brought back together. These weak hydrogen bonds during the time when the DNA was not being actively copied could simply hold it together and protect that base order. And when it was actively being copied, could easily be pulled apart so the information was fully accessible. All of these different scientists working together over all of these years put together the information that was necessary to figure out the structure of DNA.